to thank everyone for joining today's webinar. We are very excited to host Disha Moholkar, who is a third-year graduate student pursuing her PhD degree in pharmacology and toxicology at the University of Louisville. She has a master's degree in nanoscience and biotechnology from India. She is passionate about revolutionizing cancer therapeutics, especially in targeted drug delivery systems. She's currently working in the lab of Dr. Gupta, who has pioneered milk exosome technology. Her PhD project focuses on leveraging functionalized exosomes for targeted drug delivery against lung cancer. Today, she will be presenting some data from her lab focused on delivery of small molecules, RNA and DNA using bovine milk and colostrum derived exosome technology. And with that, I'd like to pass it to Disha.
Thank you so much for that nice introduction. I would like to thank everyone for joining today for this webinar. Today, I will be talking about one of the project works that is going on in our lab. And the title for today's webinar is Exosome-Mediated Delivery of Small Molecules for the Development of Novel Cancer Therapeutics. Having said that, we will be focusing on exosomes as the delivery vehicles. And as Dr. Dib has mentioned, our lab is the pioneer of milk exosome technology, which has been isolated from bovine milk or the colostrum. Today, I will be covering the brief topics such as drug delivery systems, which I will mostly focus on the lipid-based drug delivery systems. I will spend some time talking about the exosomes, which are emerging nano-drug delivery vehicles, and the focus of our research group, which is the bovine milk or the colostrum-derived exosomes. Then I will briefly talk about the tumor targeting approach that we use for our in vivo studies by using functionalized exosomes, and how we validate the efficacy of our modality by using Spectral's AMI-HTX. And lastly, I will talk about the ongoing studies on delivery of P53. Before I begin with the presentation, I would like to talk about the focus of our lab, which is developing treatment strategies against different cancers based upon milk exosomal technology for the delivery of small molecules and biologics. When I say delivery of small molecules, this mainly depends upon the vehicle or the carrier which is used to deliver the chemotherapeutics or the drug of our interest. Before I jump into the exosomes, I would like to mention briefly about different drug delivery systems which are used for this purpose. And in the literature, there are numerous different drug delivery systems which include metallic and inorganic nanoparticles, polymeric nanoparticles, quantum dots, and a dozen of lipid-based nanocarriers. However, the main criteria for being an effective drug delivery system is being biocompatible and having minimal side effects. The one from this list which closely meets the criteria for an effective drug delivery system is the lipid-based nanocarriers. And when I say lipid-based nanocarriers, again, if we jump into that, there are a dozen of different subcategories in this. And the advantages for these include being biocompatible and biodegradable because their structure is mainly based upon different kinds of lipids. There is ease of scalability because the lipids that are used for the synthesis or the formulation of these nanocarriers are widely available by different commercial companies. And these provide controlled release patterns of the moiety entrapped in these lipid nanocarriers, as well as there is ease of functionality, which is the surface of these nanocarriers can be functionalized or can be modified according to the needs of the target organ. However, these include some disadvantages such as the upscaling of these formulations would require sophisticated instrumentation, which is often very expensive. These also provide low drug loading efficiency, and the choice of appropriate lipids plays a very important role, and it is a deciding factor of what chemotherapeutics need to be loaded on the lipid nanocarriers. These also bring in stability constraints, which brings in the possibility of drug expulsion through these lipid nanocarriers. Even though there are different lipid nanocarriers known in the literature, Only liposomes, which have been in literature for the past 60 years now, have a few marketed drugs for the use for the patients. One such example is Doxil, which is a liposomal formulation for the chemotherapeutic drug doxorubicin, which is loaded in the core of the liposomes. However, the liposomes also face many of the disadvantages as enlisted here. And to tackle this, the new age drug delivery systems, which are lipid based, are exosomes, which are relatively new in literature and have the potential to be used as drug delivery vehicles. And this is the focus of our research in the lab. Having said that, exosomes are basically secreted from different kinds of cells, and these are expelled out in an envelope from the cell, and their size is in nanometric range. And when we say exosomes, as I mentioned, these are microscopic and spherical packages, and these are secreted from the cells. And these have characteristic recognition on the surface, which can be used as tool to screen for different types of cancers or guide treatment strategies. And it can also help us follow on how the patients are responding to a particular therapy. And if we look at the expenses, an exosome therapy would cost about $5,000 to $10,000. And this is mostly due to isolation or the process of upscaling the exosomes. Looking at the exosomes, these are naturally occurring biogenic nanocarriers. These are loaded with protein and nucleic acid in their core. As I mentioned on the previous slide, they are in nanometric range, specifically from 30 to 150 nanometers, and these play an important role in cell-to-cell communication. As their surface is loaded with numerous protein components, these help in natural trafficking. Due to their structure, we can either load the chemotherapeutic agent inside the core or on the surface of these exosomes. And these can be isolated from a different number of sources. Exosomes are secreted by different bodily fluids such as milk, urine, plasma, or serum. Most of the labs which are working with exosomes rely on culture media for the isolation of exosomes. Most of the labs are which are working on exosomes focus on cell culture media for the isolation of exosomes, and we are focusing on milk, which we are getting from the bovine source or the colostrum source. This is majorly due to being biocompatible and a scalable and safer source of exosomes. This would solve the drawback of other lipid-based nanocarriers by being biocompatible and having a scalable source of exosomes. Having said that, if we compare the yield of exosomes, um, milk-derived or colostrum-derived exosomes have 100 to 1,000 times more yield in terms of particles per liter as compared to the cell culture media. So this solves the problem of scalability. Now we come to the part where I will talk about how we isolate these exosomes. First, we rehydrate the colostrum powder in water, and by series of centrifugation steps, first we get rid of the protein debris, then the large particles and microvesicles. Ultimately, we subject the supernatant to final step of centrifugation wherein we get the exosomal pellet. After discarding the supernatant, the exosomal pellet is resuspended in PBS and stored in -80 degrees. We have checked different batches of exosomes for the presence of exosomal markers on the surface, and all of the tested batches were consistent in terms of the expression of exosomal markers on the surface. We further checked the size of exosomes by zeta sizer, and we found that the size of our exosomes was in the nanometric range. And this has been consistent for different batches over years. Now we come to the part where we load small molecules or nucleic acid. For loading small molecules, we incubate the small molecules or chemotherapeutic drugs with exosomes and then get rid of the unbound drug by ultrafiltration technique. And as we load the nucleic acid. Firstly, I would like to talk about what we rely on for for loading nucleic acid onto the exosomes. For this, we rely on a polycation, which is a polyethylene imine, and we call this technology as EPM, wherein E stands for exosomes, P stands for the PEI, which is the polycation, and M stands for matrix. So the PEI, the positive charge of PEI would bind the negative charge of exosomes and the nucleic acid together. And this is the base for the EPM technology. This is how we use PEI for loading of the siRNA. We have achieved high entrapment of siRNA, and for this, we relied on loading the PEI onto the exosomes and then incubating with a P32 labeled siRNA. We then check the entrapment by using a radioactivity assay, precipitate this complex by using PEG, and the pellet would have the formulation loaded with siRNA, while the supernatant will have the free siRNA. And if we check the radioactivity present in the pellet as well as supernatant, we can get an idea about how much of the siRNA is loaded onto the exosomes. And we could observe that for all the tested concentrations of PEI and exosomes, we could achieve higher siRNA entrapment as observed by higher radioactivity in the pellet. Further, we check the high stability of our formulations, wherein we relied on the RNase treatment for the EPM formulations, which was to check the degradation of siRNA, which was loaded in the EPM complex. So when the exosomes PEI and the siRNA, which was tagged with P32, and free siRNA tagged with P32 were treated with different concentrations of RNAs, we could observe the degradation of siRNA by MSA. So we can see here that when the siRNA was complexed with EPM, there was no degradation at the tested concentrations of RNAs. While for the free siRNA, there was degradation of siRNA. This suggests that our EPM formulations are protecting the siRNA against degradation. Similarly, for the small molecule, I would like to mention one example of curcumin, wherein we stored the formulation for 6 months and the anti-proliferative activity of our formulation was constant, was similar, even when the formulation was freshly made versus when the formulation was stored for 6 months. This suggests that the exosomes would enhance the stability of the small molecules as well as the siRNA. Moving ahead, where exosomes stand in comparison to its synthetic alternatives, we compared the efficacy in vitro studies as compared to its synthetic alternatives, PEI and lipofectamine. We observed that when our EPM formulations were loaded with siNerv2 as a nucleic acid candidate, we could see downregulation of NERV2 as well as its downstream effector HO1. When we checked for the PEI, which was an important component in our EPM formulation, there was no knockdown of NERV2. This suggests that exosomes play an important role in entering the cells. While at the tested concentration in our lab, we could not see a knockdown for NERV2 as well as its downstream effector for the lipofectamine, which is a liposomal formulation as available in the market. So it was clear that EPM formulations stood out as compared to its synthetic alternatives. Further, now we wanted to move ahead in our in vivo studies. For that, we wanted to have a tumor targeting approach. For this, we relied on the overexpression of folate receptors at the tumor site. To create this, we tested the expression of folate receptor, which is FR alpha, in different lung cancer cell lines in vitro as well as in vivo in the lung tissue as well as the tumor tissue. We could see that there was overexpression of folate receptor in the lung cancer cell lines as well as the tumor site. but not at the adjacent lung, which was a healthy tissue. So this suggests that we can use this approach as a tumor targeting approach. We have also validated the overexpression of FR alpha in lung tumors. So we functionalized our exosomes by using the ligand for this receptor, which is folic acid, by using activated folic acid. When we load activated folic acid onto the exosomes, we could see that there was not much increase in the size of exosomes, and it was still within the nanometric range. Next, we moved ahead to establish our tumor model. For this, we used non-skin mice and we injected A549 luciferin cells in the mice, and we imaged the animals by using AMI HDX instrument to monitor the luminescence intensity. We injected different number of cells and monitored the tumor growth in terms of the luminescence coming from the cells, coming from the tumor cells over different time points. This helped us establish a appropriate tumor model for our in vivo studies, and we relied on 2 million injection of 2 million cells for our future studies. This experiment helped us create a base for all the in vivo studies in terms of measuring the efficacy of our formulations in a tumor model. Next, we went ahead and validated our tumor targeting approach uh, for exosomes and small molecules. For this, firstly, we administered exosomal formulations and functionalized exosomal formulations to the animals, and then did the in vivo imaging, ex vivo imaging of different tissues. Here we can see that um, for the tumor tissue, there was higher accumulation of the formulation at the tumor site with the functionalized formulation. So this suggests that our tumor targeting approach was working. So we went ahead and then we loaded paclitaxel onto the exosomes. In this study, we injected paclitaxel, which was given intravenously, exopaclitaxel, which was given orally, as well as intravenously. In this study, we administered the vehicle control, which was the FA exos. And to compare the effect as compared to a standard of care drug, which was Abraxane, which is marketed formulation of paclitaxel, which was given intravenously. After this, we monitored the uh, tumor growth by monitoring the luminescence intensity over time by imaging the animals. And here we could see that vehicle control, which was the FA exo, did not have uh, had only a substantial amount of decrease in the tumor, even when there was no drug administered with it. And with paclitaxel administered intravenously, we could achieve inhibition in tumor up to 76% at the endpoint of the study, which was enhanced by using Exopac at the same concentration but orally. When we attached the targeting ligand, which is folic acid, the tumor inhibition was then further enhanced up to 45%. The notable thing here to observe is that when FA-exopaclitaxel was administered orally, it could somewhat achieve similar degree of tumor inhibition as achieved by Abraxane when administered intravenously. And when our formulation FA-exopac was administered intravenously, it could perform even better than Abraxane by inhibiting the tumor up to 24%. Further, we then validated the tumor targeting approach of EPM formulations. For this, we could see that as we observed for the exosomes, even for this one, we could achieve higher tumor accumulation by using functionalized formulations of EPM. We could see that when the formulations were administered intravenously, after 4 hours, there was higher degree of accumulation with the functionalized formulations, be it FA exos or FA EPM formulation. Also, we observed that at the tumor site, these formulations stayed up to a longer time, and while these were cleared from other tissues such as liver and lung over time, but these formulations, due to their functionalization, stayed at the tumor site up to a longer time. Then we moved ahead to the in vivo efficacy study, wherein we administered FAEPM as our vehicle control, and we could observe that our vehicle control inhibited the tumor growth as we saw in our earlier study, and the effect was enhanced by using the EPM formulation, which was further enhanced by using the functionalized EPM formulation. We could also see knockdown of KRAS at the tumor site by administering the FAEPM formulation. As we tested PEI 60K for our previous study, we also wanted to check the effect of lowering the molecular weight of PEI from 60K to 25K and further to 10K. We tested different amounts of these PEI for our next in vitro study. Here, we observed that PEI 25K, when integrated into our EPM matrix, could achieve a similar and higher entrapment efficiency as achieved for 60K. We then moved into in vitro studies where we tested different concentrations of PEI 60K and found that only different amounts of PEI 60K, and we could observe that at the highest tested amount, which was 40 microgram, there was a knockdown of KRAS, but it was not that prominent at the lower tested amounts. Similarly, when we tested the effect of different amounts of 25K, which has earlier shown higher entrapment efficiency, we could see that for all the tested concentrations, there was a knockdown of KRAS. Then we went ahead and tested PEI 25K for our in vivo studies, wherein we observed a significant reduction in the tumor growth, and this was evident by the imaging that we did over time by using Spectral's ME-HTX imager. Now I will talk about one of the ongoing studies in the lab, which is EPM-mediated knock-in of P53. For this, we have observed that even different tested concentrations, EPM formulation by itself inhibits the tumor growth of antiproliferative activity of lung cancer A549 cells by up to 20%. Upon loading with P53, it shows a similar trend. However, when we combine it with standard of care paclitaxel, there was a significant enhancement using EPM formulations loaded with P53 in combination with the paclitaxel. And this suggests that we can enhance the effect of standard of care drugs by using formulations as an helper treatment therapies. I would like to end this presentation by mentioning about few key takeaways, include that the bovine milk and the colostrum exosomes prove to be an abundant, scalable, and biocompatible source of exosomes. The colostrum exosome-based EPM technology provides significant gene knockdown, and as our recent experiments in the lab have suggested, even knock-in of P53, as well as in vivo studies, lung tumor inhibition by using targeted delivery of small molecules and siKRAS. And lastly, ME-HTX is an important tool for monitoring the tumor growth over time, and it has helped establish tumor model systems and help to image the efficacy of our formulations over time. Lastly, I would like to thank Dr. Ramesh Gupta, my PI, and Dr. Farooq Akil for being my mentors in the project work, and I could get to learn a lot from them throughout my time in the lab. I would also like to thank Dr. Raghuram Kandamalla for his helping me with different experiments in the lab. And lastly, I would like to thank 3P Biotechnologies, who helped us provide the colostrum exosomes for our studies. And with this, I conclude my presentation and thank you for your attention.